Great. Okay, so hello everyone. First of all, thank you, Christy, for the introduction. I would also like to thank the Twist team for this opportunity to talk about the Amyris technology platform. Today, I'll be presenting the journey that Amyris has taken from a company that started with the goal of producing a reliable supply of a key antimalarial drug to a company that currently has its products in more than 2,000 brands across the world. So this first slide encapsulates what Amyris does at a high level. We engineer microbes that consume sugar and convert it to pure ingredients. Our goal is to make clean manufacturing powered by biology the industry standard for how all product ingredients are manufactured in the future. Amyris was founded based on a project that started at UC Berkeley in Jay Kiesling's lab, and it was a project to develop a fermentation-derived source of the antimalarial drug artemisinin. Until Amyris came along, the only source of this drug was the cultivation of the Artemisia annua plant, which unfortunately has a 10 to 12-month growing cycle. This long growth cycle resulted in huge fluctuations for this drug in terms of supply volume and hence pricing. The Gates Foundation hence decided to fund the development of a commercial scale process where Amyris developed a yeast-based fermentation process that produced artemisinic acid in 2 weeks, which could then be converted in a few weeks to artemisinin using chemical synthesis. The result of this project was stabilization of the price and supply of artemisinin on the market, which was extremely important for developing countries, especially in Asia and Africa. This was, of course, a non-profit project for us. Sorry, that was a little slow. The result of this project was stabilization of the price and supply of artemisinin. Now, this was, of course, a non-profit project for us, but we were able to apply this technology to other products as well. We are now focused on 3 main markets, the first one of which is clean beauty, and that includes our Biossance brand, as well as products that use a squalene ingredient. I'll talk a little more about squalene in a couple of slides. But we sell our Biossance brand directly to consumers through Sephora, as well as our own website, biossance.com. We also sell squalene to other businesses through our distributors, as highlighted on this slide. The second market for us is in health and wellness. This includes multiple vitamins that are derived from fermentation and our soon-to-be-launched sweetener, as well as human nutrition ingredients, which includes our applications in the pharma space as well. Our partners in this market include DSM, Givaudan, and Nenta. Finally, the third market that we operate in is in the flavor and fragrance market, where we work with companies like Firmenich, Givaudan, and Takasago. In each case, these companies have a well-established presence in the marketplace for these ingredients, but are trying to solve some supply chain issue, whether it be cost, sustainability, or purity. And in every case, Amyris is able to perform all the R&D required to develop the strains and a new manufacturing process, manufacture the ingredient, and finally deliver the same product that gives our partners an advantage in their marketplace. So what advantages does an Amyris produced ingredient offer to its partners? First, because we can tailor the strain and fermentation conditions precisely, we can produce the same molecule at much higher purity than what might be obtained from a petroleum or plant or animal source. Secondly, we can manufacture the same molecule at a lower cost so that the benefits of a sustainable product aren't just available to people who can afford them. We believe that high-quality products should be accessible to all consumers. And finally, fermentation is simply a better process than current manufacturing practices, which include the use of petroleum feedstocks or involve the harvesting of ingredients from plants or animal sources. Let me walk you through some examples that will help illustrate these points. So let's start with an example of a better product and better purity. Many of us have used stevia as a sweetener. While it works really well as a sweetener, there's no escaping the bitter aftertaste that is synonymous with stevia. The reason for the bitterness is that the extraction process for the sweetener results in a mixture of molecules, out of which the active ingredient, Reb M, is only 1% of the entire mixture. The rest of the molecules in the mixture result in the bitter aftertaste. At Amyris, we have developed a fermentation process where we are able to precisely tailor the strain and the process to make greater than 95% pure Reb M, resulting in a very clean, flavor-free free from any aftertaste. Let's look at an example of better cost. The production of vitamin E has for decades relied on long and complex chemical synthetic route. Amyris produces a molecule by fermentation called farnesene, which can be converted to multiple products. Our partner Nenter figured out a process where the fermentation-derived farnesene can be converted to vitamin E using a far simpler chemical process that reduces manufacturing costs by 35%. Finally, a few examples where solutions developed by Amyris are simply a better process for the planet. We produce a fragrance ingredient for Firmenich, where the original source was a native plant, and 1 acre of the native plant produced about 6 kilograms of the fragrance. The same acreage of sugarcane results in 400 kilos of the identical molecule. We see similar results for a molecule that we produce for Givaudan, where we see at least 200-fold efficiency in land usage. Finally, we can't talk about sustainability unless we talk about squalene. So squalene is a molecule that is quite ubiquitous in cosmetics as an emollient. Squalane is produced from squalene, which is a biological molecule made by most living cells. Till about 8 years back, most of the squalene for the cosmetics industry was extracted from shark liver and converted to squalane. Every year, 3 to 4 million sharks were killed for this market. Amyris developed a process where we were able to convert fermentation-derived farnesene into squalene and were able to achieve purity that was the same, if not better, than shark-derived squalene. By our estimate, 1 square meter of sugarcane saves about 10 sharks, which is a testament for how biotech can offer a truly better solution for our planet. To date, Amyris has manufactured 7 different molecules at scale and currently has 12 other molecules in commercial development. Our goal and track record has been to commercialize 2 to 3 molecules every year. So how did we achieve this kind of track record? Well, with a lot of investment in R&D. We invested close to $500 million in developing a world-class strain optimization and process development pipeline by focusing on 3 main areas. The first is cellular programming, where we have optimized the process of assembling large DNA cassettes and integrating them into microbial genomes in order to introduce new functionality into the production organisms. We have also developed novel techniques to introduce entire pathways into a strain in a single transformation step. We also applied classical methods like random mutagenesis and adaptive evolution to find new strain lineages. Overall, we send about, I would say, 1.2 million strains per year for analysis, which then highlights the next area that we had to spend a considerable amount of time and effort in developing, and that is analytics. It's of no use to create so many strains if you can't measure the impact of the cellular changes. We have hence developed methods to not only analyze the extracellular molecules produced by the cells, but to also quantify the intracellular metabolites and proteins. We believe that it's important to not treat the cell like a black box, but to instead use it to expand our understanding of cause and effect so that the next molecule we work on can benefit from the lessons learned of the previous one. Our current pipeline can analyze to the tune of about 10,000 samples a day across multiple measurement platforms. Finally, as you can imagine, building so many strains and analyzing so many samples results in a huge amount of data that quickly becomes overwhelming for any human. We have hence developed sophisticated computational metabolic models that we can overlay with the data. We apply proprietary statistical models and machine learning algorithms to these metabolic models to understand the impact of the cellular changes and to drive the next set of decisions. As our data set increases, it will drive the next set of improvements faster, resulting in fewer and fewer iterations through the strain engineering cycle. All this investment has culminated in a fully integrated strain optimization pipeline where we combine next-generation tools in molecular biology, metabolic engineering, and fermentation process development with automation and computational tools. However, what's missing with this picture is a final stage for this pipeline, and that is manufacturing. Because of Amyris' experience in manufacturing 7 different molecules with varying physical and chemical properties, Amyris has learned some hard lessons in the difficulties of scaling lab-based results to predictive performance at the manufacturing scale. Unless your entire R&D machine takes into account the final manufacturing requirements of your strain, even the most sophisticated pipeline will fail to deliver the final strain and process that performs as required at scale. Finally, We rigorously capture data gathered at every step in this pipeline and feed it back into the design process with the goal of continuously improving the pipeline and our understanding of strain physiology. I also want to call out the fact that we shouldn't discount the impact of human intelligence in this step. So while we do apply a huge number of computational tools to understand what's going on with the strain, we also have humans who curate this data and then try to understand what they should be doing next with this. I would now like to spend the next few minutes discussing some of the computational tools that we now apply to elucidate the most optimal pathway to a certain molecule, and design tools that enable scientists at Amyris to rapidly and without errors come up with thousands of DNA designs to convert an ethanol-producing yeast strain to, say, a farnesene-producing one. So as this slide illustrates, all living cells consume sugar in some form or the other and convert the sugar into all the molecules they require for survival. In order to hijack the cellular machinery to produce a target molecule, you hence need to first figure out the reactions needed to convert sugar to, say, a molecule like farnesene. We had then built a database or living atlas of all the reactions known to occur in living cells. We have developed tools that can access these reactions and for a given molecule, map out the most optimal route to that molecule from sugar. However, this is not as trivial as I just made it sound. Imagine you use Google Maps to get directions from one location to another. The software will try to get you the most optimal route by avoiding major traffic bottlenecks or without sending you in the wrong direction down a one-way. In a similar fashion, our design tools can't just map the route with the smallest number of reactions. They have to take into account elements of thermodynamics, cofactor usage, and pathway balance to come up with the most optimal route from sugar to that final target. And so that's what this will look like. So in this case, for every, so what's important here is that the software needs to calculate what the highest possible yield from sugar to that molecule is going to be. So in this case, for farnesene, for example, for every 100 grams of sugar that is fed, about 24 grams is converted to the product. This is an important number because of what our manufacturing team does with this information is they combine it with all the operating expenses data collected from our manufacturing sites and develop a cost model to predict what kind of manufacturing costs we can achieve at commercial scale. So again, unless a company has experienced a full-scale manufacturing campaign, it's very difficult to understand just how much work the R&D team has ahead of it. Okay, once we identify the route, it's time to convert it into DNA code. Every reaction is carried out by a protein. Everyone knows this in the field. So every reaction is carried out by a protein. Every protein is encoded by a unique DNA sequence. Our design software will map out these DNA sequences associated with each reaction and combine these DNA sequences with the other DNA supporting elements like promoters, terminators, tags, etc., which are required to functionally express the required proteins. Of course, it's very rare that a single DNA code will provide the functionality you need, and typically you'll need multiple DNA commands to program the cell to carry out the new activity. This is, of course, manually very labor intensive, as anyone has ever done this will attest to. So what we've done is developed a genome specification language for our scientists so that a simple written instruction like the one shown here translates into a complex DNA sequence as shown here. Now, for anyone who's ever done this, you understand what goes through this process. It's a process that a few years back required hours of manual curation, a huge amount of copy and pasting from public databases into Word or Excel. It's a highly error-prone process. Today, it takes a few seconds. Now, even though our understanding of biology keeps getting better and better, I would argue that biology remains highly unpredictable. And even picking the right DNA sequence for a particular reaction is not a trivial exercise. So scanning as much diversity as possible for every reaction is key to finding the optimal solution. And why is that? Again, these are various reasons that I have listed here. I'm sure most of you could come up with multiple other reasons why you need to scan genetic variants. But the top two, I'll just talk about the top two. You can read through the others. The first one is what happens if the native reaction doesn't exist. So For a lot of times, the molecules we're trying to make are not made by the production host that we're trying to use. It's also possible that the native reaction does exist in that host, but the activity is just too low. And it is possible that the same reaction is carried out far more efficiently by a protein found in another organism. Another reason could be that in order to get the activity you need to manipulate protein expression to increase the activity. You can do this by varying the codon usage. Now, of course, all the other reasons, decoupling the pathway from regulatory mechanisms, minimal side reactions, cofactor dependency, protein localization, all these are factors that you have to take into account. However, the only way to bring together this level of diversity in a host is by synthesizing the various genes. And this is where gene synthesis technology pioneered by companies like Twist have changed the landscape. How quickly you can make progress in any project is defined by how many shots on goal you can take in the experiments. And so over here, I show the design, build, test, analyze cycle that everyone goes through when we have to optimize a strain to make a single molecule. Now, like I said, how quickly you can make progress in this design, build, test, analyze cycle is how quickly you can go through it and how many times you can do it per unit time. The more high quality shots you can take, the higher your chance of actually scoring. The cost of taking the shot, the amount of time it takes, and the quality of that shot all impact the likelihood of success. Now, I'm sure a lot of you can attest to this, but back in 2003, I used my first synthetic gene. It cost me $3,200, or actually it cost my PI $3,200, and it took 3 months to synthesize a gene that contained multiple errors, which I then had to correct later on. I had to pick one candidate for this reaction, and I had to be either absolutely sure or really lucky that this was the right gene to pick. Today, if I had to do the same, and I had $3,200 to spend, I would be able to test 20 candidates in 1 month without worrying about the quality of the experiment. And this is how Twist has changed the game. You don't need to try and pick the right candidate. You simply test them all and reduce the chance that you missed anything. Now, let me show you some actual data from a screening experiment we ran at Amyris a few weeks back. We were working with a 7-step pathway and screened multiple genes for step number 5. As you can see, the results vary significantly from one version of that gene for the same reaction to the next. If I had only one gene to test, I would need to be incredibly lucky to not pick gene 4 or to pick gene 10, which are the lowest performers and the highest performers in this data set. But this is the advantage that cost-effective, high-throughput gene synthesis brings to companies like Amyris today. It takes the guessing out of the equation. You can test a huge amount of variation and pick the right one based on actual empirical evidence. Now, this is where a company like Twist really gives us an advantage. Now, let me finish up last few slides by tying all this back to our ability to predict performance at manufacturing scale. And this is by focusing on data quality and prediction of strain performance at the lab scale. When you need to test 1.2 million strains a year, okay, you can't possibly test them all in fermentation tanks. And yet that is exactly what you need to do to predict manufacturing, a manufacturing process. What we've done instead is we've developed culture models in 96-well plates and used them as surrogates for predicting performance at the larger scale, understanding fully well that there are differences between the 96-well plate model and the fermentation and the half-liter or 2-liter fermentation scale and the manufacturing scale. In order to account for these differences, what we've done is develop algorithms that pick winners from the plate models that are then tested in lab-scale fermenters under conditions meant to reproduce what the strains will experience at manufacturing scale. Again, because of our experience in scaling up strains and processes, we've developed a lab-scale fermentation pipeline that is capable of perfectly predicting performance at manufacturing scale. This graph represents different strains for one product that was sent to manufacturing over the years. And in every case, the lab performance has matched performance at manufacturing scale. This level of reproducibility dramatically reduces risk at manufacturing, where every error is risky. And this reproducibility also shortens time to market. Let me now show you a quick video. Let me show you a quick video of what a large-scale fermentation facility looks like. This is a fermentation facility that we operated in Brotas, Brazil. It sits next to an ethanol mill that we buy sugarcane juice from. These pipes you see are transporting the crushed sugarcane juice from the ethanol mill over to us. And let me see if I can go back a little. And here you can see the crushed sugarcane, which is known as bagasse, piled up here. This bagasse is burned to generate electricity that runs the entire facility and also supplies power back to the grid. This is a bird's eye view of the 6 fermenters along with the storage tanks, chillers, and compressors. So we have 6 fermentation tanks at this facility. Each fermentation tank has a volume of 200,000 liters, resulting in a working volume of about 1.2 million liters per fermentation run. We also utilize multiple contract manufacturing groups to accommodate the different molecules that we produce, and are currently in the process of building 2 new manufacturing facilities as well. So, in summary, over the past 10 years, what Amyris has done is built the first fully functional automated organism engineering pipeline. This has resulted in a 10-fold lower development cost for new molecules and a 5-fold reduction in development time. We have successfully, to date, commercialized 7 target molecules and produced them at the kilo to ton scale. We currently have 12 molecules in the development pipeline and expect to launch 2 to 3 molecules every year for the next 3 years. And I think finally, it's important to note, low-cost, high-throughput, high-quality DNA synthesis has been a game changer for the industry, reducing risk and uncertainty in optimizing strain performance. So with that, I would like to thank you for your attention. I'm going to now hand this